Let's start. So today it's our pleasure. So we have Carolina Wählby as a speaker. So she is not working at EMBL, but we just asked her whether she would like to present in this Janelia EMBL bioimaging seminar series. So I know Carolina already quite a while. Maybe most of you too, but I will just say a few words. So she is a professor at Uppsala University, interested in quantitative microscopy and also including spatial transcriptomics, which will be the focus of her talk today. She also is the scientific director of the so-called SciLife Lab. So this is actually an initiative I have quite some connections with because they are also focusing on providing bioimage analysis as a service, so which is also my job. So there's Anna Klemm in this group, who some of you might know, who I collaborate with quite a bit in this respect. Then something else, which maybe I don't know if all of you know, but she is in some other way also very famous because she was a co-developer of Cell Profiler at the Broad Institute for 6 years, from 2009 to 2015. So I was a big Cell Profiler user and fan. So thank you, Carolina, for helping there. And then something else I wanted to mention because I thought it was quite impressive. So Sweden actually, or specifically the Knut and Alice Wallenberg Foundation, decided to really invest into data-driven life science, and they have a 300 billion euro effort to move this forward. And there are many positions open, I think, and many interesting initiatives. And actually, Carolina is a member of the steering committee of this effort. All right, that's, I think, what I wanted to say. And then Carolina, thank you very much. And we are looking forward to your talk.
Thank you. And thanks for the nice introduction. And yeah, about the open positions, there will be, formally, we were not going to have more group leader positions, but now there will be a few more coming up in a half year or so. So if you're interested in this interface between life science and computations, there are more possibilities coming up for nice and well-funded group leader positions. So very nice to be here. And as I mentioned briefly, my computer crashed like 10 minutes ago, and I had to restart it. So I don't know if my last edits to my PowerPoint really got stuck, but it might be a little bit messed up, but I hope you will bear with me. So now let's see if I can just move forward among my slides. I kind of want to start in the end and say why we want to use this type of methods, this in situ sequencing and spatial omics. And this is some collaborators that approached us with this problem some time ago. And this is about understanding the tumor microenvironment, and more specifically in colorectal cancer, when there is a liver metastasis. And it turns out that as a liver surgeon, you usually see more or less two different things. Either you see that the tumor cells have infiltrated and sort of replaced the liver cells. So all the different cell types are kind of intermixed. And the tumor cells are not very well differentiated. They are dividing a lot, and they're quite motile. And it's very, very difficult to remove the tumor without more or less destroying the liver. But then there's also the better scenario, and that's the desmoplastic growth pattern. And in this case, the tumor cells are more differentiated. You can almost see the intestine kind of style of growth pattern here. And then this is the normal liver. And in between, we have this rim. So the liver has sort of encapsulated the tumor. And this means that it's surgically much, much easier to remove without harming the liver. And then the question now is what is the transition between these two? And it actually turns out that it seems like this pattern appears first, and then in some patients, this pattern comes later. So it's some kind of reaction by the liver. And then the question is, can this more favorable growth pattern be induced by therapy? And could there be novel targets for oncological therapy and that could sort of improve the survival chances for these patients? Can we make the liver sort of understand that it should do this encapsulation so that we can remove the tumors? And to understand this, we need to understand what's going on in this rim. We need to know what kind of cell types are there and what they're doing. And this is where in situ sequencing becomes really, really useful because we can look at many, many different things at the same time. So if we compare to other methods, we can, of course, just look at the morphology and from that conclude that we have these two different patterns, but we don't really know anything about the characteristics of these cell heterogeneity. We can do bulk sequencing, and we might then find differences in gene expression, but it's also difficult to know if those genes are really involved in what's happening at the rim or not. We can do single cell gene expression and know what cell types are in the mix. But if we don't know where they are, we can't say so much. And this is where we really want to have this in situ sequencing to know what is happening where in this interface. And yeah, some history. I've been working with counting dots for many, many years. This is my first paper on counting dots from '97. My last name was different back then. But this kind of has continued until now. So that's what I do for my research, count dots. I did some more, also some cell segmentation. This is actually what went into Cell Profiler. That's finding cells in tissue. But then we started to look at these different ways of decoding or counting dots, where each dot would represent an amplified copy of some specific gene. We started doing it on glass surfaces, but then that's when I was doing a postdoc with Mats Nilsson's group. And then he kind of came up with the idea that maybe instead of having all of the RNA in a solution, we could actually find it directly in the tissue sample. And this is what we published in 2013. And it was a way to sequence RNA directly in the tissue sample. And already back then we were looking at tumor data. In this case, it's a breast tumor. And I thought that I'd just briefly go through how this version of in situ sequencing works. So my talk in general is to sort of go through this in situ sequencing principles and different approaches that we have used for decoding. And then I wanted to focus a little bit on what we can actually learn from in situ sequencing data that we didn't really know from other methods from before. And then finally talk a little bit about how we can go beyond just a human analysis of tissue and how we can use in situ sequencing together with tissue morphology. So that's my plan. And I need to move this to also keep an eye on the time. So I will leave like the last 10 minutes or so for discussion. That sounds okay. Good. So the original in situ sequencing method that we developed had started with a reverse transcription from the mRNA to cDNA. And then we work with padlock probes, which means that you can either search directly for the code that you know should be present, or you can search for the start and the end of a specific code, but not know what is in this gap. And if you do this version where you have this gap fill approach, you can find like single nucleotide polymorphisms and so on just by filling the gap. so that the unique code that you later want to decode is what was actually the complementary code to what was in the sequence here. Or you can have this targeted approach where you design like a barcode. So whenever you get this circle here, you know that the probe that had this particular barcode found its target. And in both cases, you get circular DNA in the end, and then you can run rolling circle amplification so that you get many, many copies that are then stuck to the tissue exactly where the detection was done. And then if we zoom in on this, you have the barcode there or what is in the gap filling. You have an anchor primer and you have sequencing probes, and then they will bind depending on what sequence you had present. And then since it's many, many copies, the whole blob turns green in this case. And this is what it would look like in the tissue. And this can then be done on large tissue sections so that you can get a lot of information across the whole image. And then if the first letter was a C instead, you would get a pink signal and then a pink dot in that corresponding position. So then you would go on like this and sequence the first, second, and so on, as many positions as you have. And then you have this data set that one image for each hybridization step, and then to know what gene was present or expressed in that particular position, you can do the decoding. And since you know it's, if you have designed these barcodes, you would know then what this particular code would represent. And you probably know this from before, but then if you have length of barcode length of 4, you can find as many as 256 different barcodes at the same time. And if you increase it to 5, you can search for more than 1,000 different barcodes at the same time. And this is then happening at the subcellular resolution. These are 2 cell nuclei. So today there are many, many more image-based in situ sequencing methods. I think what's kind of unique for our approach is this rolling circle amplification that makes sure that the lobes are large enough to be possible to find by regular fluorescence microscopy, which means you don't need to have any specific high-resolution imaging technique to find them. But all of these different methods are conceptually very similar. You have large image series, and you have some kind of combinatorial labeling of the different RNA species. So why would you want to do some kind of in situ sequencing when you could just as well use multiplex fluorescent staining instead? I mean, with a multiplex fluorescent staining, you would see the proteins, and the proteins might tell you more about the true action that's going on, rather than just looking at the RNAs. And there are 3 good arguments, in my view. I mean, of course, the methods are complementary, but one argument is that you can have this multiplexity. You just have to do 4 or 5 cycles to look at 1,000 different things. That's not possible with immunofluorescence staining. And then what I will get back to a little bit later is that in the case of in situ sequencing, what you always do is to search for point signal. You're looking for something that is point shaped. And that means that you can guide your analysis both by intensity and shape. When you have immunofluorescence staining, you have very different shapes for very different objects, and it's often very difficult to know what is a true signal and what is background. While with in situ sequencing, you know that if it's something that is dot shaped, it should be a true signal. And then also, I mean, it's a targeted approach. You know what you're searching for, or you need to know ahead of time what you're searching for, so you can avoid sort of housekeeping genes and things that are present everywhere, and focus on what you really think will differentiate the cell types that you're interested. And then also, proteins degrade with different speeds, while mRNAs have more similar stability. I mean, it of course depends on a lot of different things, but it seems like the RNA is a bit more stable than many proteins, or the variation in protein stability, which means that you don't have that bias that makes different samples look very different just because they have been out on the bench longer, or being exposed to light or whatever. And yeah, so as I mentioned, the fact that you know that you're looking for a round signal means that you could do simple filtering to really enhance only the signals that have the shape and the intensity that you're looking for. So you have both the shape and the intensity to search for when you decode. I also want to mention briefly that using this idea of having circular DNA and doing rolling circle amplification and multiplexing can also be used for detecting proteins by dual binding or protein interactions, or in this case, the binding of a receptor and a target. So the same kind of idea of getting a signal whenever you have two pieces of RNA making a circle is useful also for other types of detection. So when we decode the data, there are several different challenges. We can have limited signal-to-noise ratio, background fluorescence, some jitter or some movement of the signals. We can have very dense signals. We can have poor alignment across the sequencing cycles. And we can also have some of these RNA products or dots being a little bit spread out. So this means that we have to think a little bit about how we decode the data. And also, there's a challenge to do the evaluation, because it's difficult to know if a signal is a true signal, and if the pattern or signal barcode, is it expected in the code book, or is it a signal that just appears by random? And we have been working with many different approaches for decoding. And there are lots of different code out there for doing it. But she specifically asked me to mention Istico. So I thought that I'll just go through the different barcoding or decoding approaches and tell you about this method that we call Istico. So most methods have the approach to first kind of find the dots and then decode. So you would have either some kind of anchor probe that binds to everything, or do some kind of projection across the different cycles, and then find the dots in that merged image. And then you would measure in which channel you have the most highest intensity, and then you would assign the corresponding letter to that. And that is sort of the most common approach to decode these. This is another approach where you would have like a graph-based approach. So you would think of sort of going through the cycles and finding the best path. Where would I have the most similar signal across time? But then what I want to talk the most about today is actually where we first decode the pixels and then find the dots. And this is the method that we're using the most today. So if we look at this, and if we first think of what is the data that we have. So we have the in situ sequencing images. In this case, we have 4 channels and 4 rounds. And we could also, so we can represent this as a 3D volume where we have, if we zoom in a little bit here, we have the sequencing rounds, and then we have the different colors. So if we would cut out a slice from this 3D volume, it would look like something like this. So in the first, the slice that we have in front, we have one red and one blue dot. And then in the next round, it's green, blue, red, red, blue, red. So if we would decode it, this is what we would get. Now, you should notice that you can usually also capture the data as multiple focal planes. So it would actually then be 4-dimensional. And the decoding that I will present works sort of the same also if you would work in the 4 dimensions, but it's more common to project it into 2D, as you usually work with rather thin slices. But if you work with more, where the 3rd dimension matters, you can use these decoding approaches in the 3-dimensional space. So if you think of these different approaches where you first find the dots and then decode, they all have a way of finding the dots and then doing some kind of nearest neighbor matching. So you would find the dots. Here is one dot and here's a second dot. And then once you have one of the dots, you have the intensities across the rounds and channels, and then you have your code book, and you would match it. And then you would say, okay, this particular dot belongs to this position in the code book. And here, where the dots are fairly sparse, that works well. And this is a good and efficient way to go. But if you have a lot of signals and optical crowding, the corresponding sort of data might look like this. So you have signals that are very much overlapping, but at the same time, all the information is there. So if you would just find the maximum intensities here, you would lose a lot of information. So let's say that you assume that this is where you would have a maximum intensity and therefore a dot. And then you search for this signal in the code book. You don't know which one of all these it would be. But since we can see here that the signal that we see in this round, in this position, is probably something that belongs to a different signal than the signal that we're currently looking at. So we have the information to sort of decipher which signal it comes from. But if we first find the dot and then match with the code book, we have lost that information. So therefore, we instead want to present this image data as a combination of many different things. And one is the sort of crosstalk or the leftover. Sometimes we don't completely get rid of all the signal from a previous round. So this would be a representation of the crosstalk or potential leftover that we would have in our data set. And then we have our code book. And then we have the actual deconvolved image that we want to find in the end. And then we also have some spatial blur. And then, of course, some background and some noise. But the nice thing here is that the spatial blur we can find from the microscope, the code book we know, and the crosstalk we can also find from our data. So then what we really want to find is our deconvolved image. And then what we can do is to work with the deconvolution and sort of search for the most likely true deconvolved image that we would have given the output that we observe. So to do this, we use non-negative matrix factorization. And I could show you all the equations for it, but I would prefer to just refer you to the paper, which is on archives by my student, Axel Andersson, who is defending his thesis next week. So the output, that's sort of the drawback with the method, is that the output, rather than being directly a map of where you have each and every signal is that you have one probability map for each gene that you're targeting. So you go from a rather compact data set to a bigger data set. And then you would do your dot detection in this data set. Of course, you don't really have to dwell with this big volume too long. You can go, directly to find the dot detection here. But at the same time, it might also then be useful to have this sort of probability map for each gene so that you can work with deciding how important it is to find every signal, or how confident you want to be with the signals that you continue to work with. I should mention that bar denser and postcode are using similar principles, but so far we have been more happy with output with this approach to the decoding. So then how do you know what is the best way to go? You have to work with some kind of quality control. And there are many types of errors. You can have biochemical, imaging, and decoding errors. And it's sometimes difficult to know which is which. And you can work with this by introducing unexpected barcodes in your codebook and see how much of the unexpected barcodes do you find, how much and how many of the true barcodes do you find. And you can also introduce in some kind of quality metric that would be based on, for example, the intensity across the rounds and cycles. And then you can see what would be a suitable threshold for excluding unexpected barcodes and only including expected ones to sort of know where the difference is between noise and true signals. This is a tool that we developed that's called tissue maps, where you can visualize your signals. And you would upload all your data from all your rounds and all your channels. And then you would look at the decoded data, which is represented by these different symbols. And then you can go in and click on a specific detected signals. And then you would see what are the intensities in that particular position across all the rounds and channels. And we found that this was really, really useful for figuring out if you have, part of the tissue where you have an idea what genes should be present. And then if you have something there that should not be in that position, this is a very efficient way of figuring out if you might have debris, or if you have something strange going on with your signals. And this is available to try on our GitHub page. What I mentioned briefly before is that one of the most tricky things that might happen is when you have poor alignment of your data. And this is where we make use of Ashlar, which we're very happy that it's available for free to align your data. So how do you choose your decoding approach? I think that it depends on the quality of your data, and also if you have sparse signals with high intensity, I would go for just finding the dots and then decoding. And that could be a good way to also do like a preliminary decoding, and then do the more advanced and time-consuming decoding once you have sort of tested the quality of the data by a fast method such as Stardist and so on. And then you can search for more signals with the more advanced method. So what can we learn from in situ sequencing data that we didn't know before? Well, if you, in general, if you want to analyze tissue samples, you usually want to understand and quantify similarities and differences in things like cell distribution, organization, interaction, and so on, and relate this to some kind of biological or biomedical question, such as this infiltration of the tumor cells in the liver. And then hopefully relate this to some kind of patient prognosis and treatment response. And then if you use in situ sequencing, we know that the mRNAs are more or less equally preserved as compared to proteins that have very different half-lives. We're searching for dots and not only intensity, so the quantification is easier. We can do high multiplexing, and we can observe the distribution of many targets in the same sample. And we can also know where we have activated genes, which is maybe then a bit of a drawback, because activated genes might not always reflect selectivity. But one of the challenges once we have done in situ sequencing is the richness of the data. This is the developing human heart, and more than 100 different genes that have been targeted here. And as you can see, we have subcellular resolution, and if we look at everything at once, we don't really see patterns, but if we choose a subset of the genes, we can clearly see that there are different things happening in different parts of the tissue. And this is where we kind of want to extract some kind of biological information. And we have really realized that viewing things together with the experts is key before you know what it is that you really want to quantify. And a lot of people say they want to count cells. But counting cells, I mean, you want to count cells because that's what you do in single cell sequencing when you work with homogenized samples. But the tricky thing with 2D slice from a 3D tissue is that you might have the ideal case where the cell nucleus is centered in the slice, but it's very often that you might have the genes, but not the nucleus, or you have very clustered nuclei, and it's really difficult to assign genes to the right nucleus. And the cells, they may be elongated, so you have a big part of the cell, but not the nucleus inside your tissue. And they also might look very different. You might have some cells that are big and some that are small. And then if you count cells, you really introduce a bias, because large cells are more likely to be sampled than small cells. So the count of large cells will be overestimated. And there is this Achille Deleuze back in the 1843. He showed that the area fraction correlates well with the volume fraction. But he also showed that the number of objects in a 2D cross-section is not related to the number of objects in 3D. And this alone, I think, is a good argument for saying that we might want to look for areas rather than numbers of active regions in our cell. So, I mean, cell segmentation is in many ways a very well solved problem nowadays, both with StarDist and CellPose. But maybe it is not cells that we want to find in the tissue. Maybe we want to find niches, regions where we have things going on, rather than knowing exactly which, how many cells we have in each position. And if we only focus on finding cells, there is a big risk that we miss important things just because the cell nucleus is not present in that particular region in the image. And if we always rely on good cell segmentation and the cells are not present, we might lose important information. So what we want to do is to sort of go beyond looking for the individual cells and instead relate the local gene expression pattern to the function. There are methods, many methods for sort of doing cell typing where you do first rely on the cell segmentation. But if the cells are dense like this, it would not work very well. So what we try to do instead is to find niches or find local similarities and differences in gene expression. So if we sort of ignore the individual cells and instead describe the local composition of gene expression. So for every dot, you would look at its neighborhood. And by doing this, create like a vector. And then present this composition vector in a multidimensional space where the space has as many dimensions as you have targeted mRNA types. And then you do clustering in this feature space. The radius that you choose will have a big influence. And if you have a small radius, you might be finding cells, so even smaller radius, subcellular niches. Or if you have a larger radius, it would be more multicellular compositions. And if we just look at, without doing any kind of clustering, and instead we take this whole three-dimensional space, I mean, not three-dimensional, as many dimensions as we have different mRNAs, and we reduce it to 3 dimensions. And in these 3 dimensions, we just present the 3 dimensions as an RGB color cube. This is what we see. And we clearly see in this mouse brain here that there are, in some cases, smooth gradients, and in some cases, really sharp transitions. And in this, then, we can do clustering and find different regions of interest. And we could even select, a few marker genes and do the clustering based on those. And in this way, automatically define the regions of interest. We don't have to manually define different parts of the tissue. And then once we have used these known marker genes, they can guide us in exploring similarities and differences in other genes to answer questions. So we have been doing this clustering for quite some time. And as I said, the radius makes a huge difference. And also other clustering parameters, like how many clusters you choose. And in order to make it possible to play with these, we created what's called points to regions, which is a interactive tool for doing this clustering. So this is also parts of our online tissue maps. So what you can do then is to go in and interactively adjust the size of the neighborhood that you're looking at and the number of clusters, so that you can play with the data and more or less automatically figure out what would be the best parameters for finding the different regions that you're interested in. And we have compared these to, again, knowing what the ground truth is always difficult, but it seems to be quite strong correlation between the cell types that we can find by this completely unsupervised clustering as compared to previously published descriptions of cell types in the tissue. And I think the biggest benefit of this is that it's interactive and that you can work with both cells and subcellular and more cell niche discovery. So if we go back to where I started, this use case of the liver, what we wanted to do first was to find these different regions in all we had 10 samples from 10 different patients. So if we look at all the different reads, we saw something like this. And then we realized that albumin, for example, is a perfect marker for defining where we have the healthy liver. And then we had 2 specific tumor markers and 2 markers that were specific for this rim region. So using these 5 mRNAs, we could automatically partition all our tissue samples into healthy liver, rim, and tumor. And this was also done manually by our pathologists, only looking at the corresponding H&E image. And there were differences, as you can see. And if we zoom in a little bit, we can see that the manual analysis is a bit more coarse, while it's more detailed lines in the automated. And then also in some cases, visually it was difficult for the pathologist to see where the border was between tumor and rim, while with the use of the mRNA or the in situ sequencing, the delineation was a bit different. But in general, the two patterns or the two different definitions of regions match quite well. I was considering showing this live. If you bear with me, I will try to get this to work over Zoom. It's always a bit scary when you can still see my screen, right? Good.
Yes.
So here are our 5 samples. And as you can see, we have the samples, and in parallel, we have the H&E staining so that you can, it's actually parallel slice because we didn't want to have any influence of the in situ sequencing on the H&E staining. But here you can see this is where you have tumor, this is where you have this fibrotic rim, and this is where you have healthy liver. And the regions about here is, now they have been just overlay of color here to show where we have the different regions that were defined by this automated detection. Let me just hide the, no, I'm on the wrong place. Let me see, 1905-16. So I'll just hide these colors to show them the raw nuclear data. So now we can show the genes on top of this. So now it's downloading all the genes for all the 10 samples. So we have, so you can sort of observe everything at once. And as I mentioned, albumin would be a good marker for where you have the healthy liver. And then what we wanted to explore, what was going on in this rim. And what we have done then is to run this kind of clustering, focusing on only the part of the data that is in the rim. And we did a joint clustering across all our patient samples and as you can see, that the sample that I'm showing here is quite nice and clean. But in other cases, depending on how the tumor has been cut, the rim and the tumor are more mixed up. So in order to do some kind of draw some kind of conclusions from all of this, we needed to do statistics across all our data sets. And it's taken some time and lots of discussions with our collaborators. But what we have done in the end is actually to find some specific regions and clusters in this rim that are statistically significant across all our 10 samples. Since the data is so rich, it's otherwise difficult to know what is really significant and what is not. And this is the type of analysis that we decided to do. So once we have found the healthy liver, the rim, and the tumor, we have been able to then look at the distance of how far from these different regions, different genes are being expressed. And to sort of figure out what signals originate from the tumor and what signals kind of come more from the rim side. And what I think is most interesting here is that we find this NGFR, which is a inducer of the fibrosis reaction, or the creation of this rim, which is much more highly expressed in a short distance from the liver. When we're close to the liver, we have a lot of this fibrotic reaction going on. And that sort of indicates that the liver is starting this encapsulation from its healthy side, while this other post-end gene is more initiated than from the from the tumor side that's highly expressed close to the tumor. So we believe that that's the signaling that is coming from the tumor to initiate the angiogenesis and making sure that the tumor gets more vessels and so on connected to it. So this is where we had the largest significance in our resulting analysis, and this sort of initiates us to target a new set of genes to figure out more about what is actually happening in this interface. Yes. So what we have learned from this is that visualization is absolutely necessary, both to ensure image quality, but also to communicate the richness and the limitations of the data with the people that are the experts in the data, the samples that we're looking at. And we need to make use of a priori knowledge of the tissue morphology and spatial heterogeneity to figure out how to do the analysis. And also, we very often have to have many iterations of visualization and so on to figure out what is actually relevant in this rich amount of information. And this is where it's been really useful to have a web-based interface, a web-based tool where everyone can log in and make annotations and look at clusterings and so on produced by others without having to send data back and forth. And what we want to do next is to go beyond what a human can do and also incorporate the tissue morphology. And we have been doing things on like automated cancer grading before. And this is kind of working well, but it's only reproducing what a human can do. Which is, of course, good, but not maybe so interesting from a scientific point of view. So we have been using these pre-trained networks then that were trained for classification to also look at clustering of the data based on morphology rather than on gene expression. And it turns out that the morphology also matches gene expression in such a way that part of the tissue that have been manually annotated have the pathologist has not seen differences in this upper and lower pink part of the tissue, while the automated analysis clearly indicates that there's different morphology, different part of feature space that's up here and down here, and that's also reflected by differences in the gene expression. And in this case, it's spatial transcriptomics that's sort of related to the morphology. I want to jump to here. This is my second to final slide. And this is where I think that there's a lot more work both going on in many labs in different places, but also where I think that we will see more things happening in the future. And then if you think of like this blue circle being all the relevant data that you can collect from a patient, it can be everything from a heartbeat, from or heart rate to disease history and so on and so forth. And then you can run different types of spatial omics. And part of what you find from the spatial omics is relevant for the outcome of the patient. Like specific genes that are related to the disease. But of course, there's also lots of information that might not be relevant for the outcome of whatever that has happened to this patient. Then you also have information from the tissue architecture, from like standard clinical staining, that also has overlap with the diagnosis for the patient. And this is the type of data that we have, biobanked and lots of collections, but maybe we haven't really fully explored this. And if you do, In this case, we used ResNet 50, and then we used the self-supervised learning to sort of fine-tune to fit the data, and then clustered on these features. We see that there is quite a lot of variation in morphology that is not super different from the variation in gene expression. So if we then just understand these differences in gene expression, in morphology, maybe by using the gene expression, we are sort of in this little overlapping part here. And that means that what we learn from genetics can also be found in standard H&E staining, which is available in a lot more clinics and also in these biobanks, so that we can confirm what we have in retrospective studies. And I think this is really a place where we can make a big difference. And with that, I want to thank you for listening. And these are members of my group that have contributed to this. And then, of course, also many, many collaborators all over the place. Thank you.
I can start maybe. I have a question, technical. What do you say, is there like a limitation with these points to regions? For example, would that also work if I have like a very lonely T cell surrounded by a lot of other cells, or is there, yeah, so I don't know.
Well, when you set the parameters, you say what the minimum signals within, you define how big your neighborhood should be, and you also say what is the minimum number of signals you would need in your neighborhood for considering it interesting to try to cluster. So then if this particular cell that you're looking for, if it has many signals, and if your radius for defining the sort of local neighborhood is in the same kind of size as the cell, I think it's quite possible to find it. Also, of course, you choose how many clusters you want, and then you would need to have as many clusters as you have cell types.
Okay, but a cluster could also have just a few cells. So the method is not enforcing sort of implicitly many cells per cluster, or is it? Or
no, because sometimes these in situ sequencing data sets are sparse, but there's still information there. So then and that's what we find kind of useful, and that's why we want it to be interactive also, because then you can still get something useful out of your data, even if you can't find things at a single cell resolution. You can still find similarities and differences across your data set. And then a particular region would be more what we would call a niche, or a neighborhood where you have a combination of genes being expressed.
Okay, thank you. Greg?
Hi, thank you for the very informative and interesting talk. So you guys have favored this decode the pixels, then detect the spots first, or then detect the spots second approach.
Yes.
But that also includes this deconvolution, which is not usually included in the detect first, then decode approach. Like, I'm not sure if Starfish includes an optional deconvolution or not.
Oh, now you're talking about the optical deconvolution.
Do this non-negative matrix factorization to optimize this deconvolved version of the image. So kind of implicitly in your approach, you've kind of made two changes from the sort of standard. You've swapped the order and you've added this deconvolution. Do you know, like, to what extent those two changes sort of relatively impact the results? Like, does one of those two changes account for the improvements more than the other?
So we would like to have the deconvolution in the sense of the full point spread function in 3D incorporated in the step as well. But what we're doing currently is to first pre-process the data with a more standard deconvolution per image round and image channel. And then we apply this sort of second deconvolution, which is deconvolving the signal composition rather than the actual blur. But it would be, if I could understand you correctly, it would be much more beautiful if they could be incorporated in the same step, which I think should be quite possible.
I guess the reason that I'm asking, I do the more forward approach, and I guess I have the luxury of really like well sampled 3D data. So for the data that I work with EasyFish data, and so it's thick slices, and we have pretty good resolution in Z, so I can get the full shape of the point spread function pretty easily from every round. And in my experience, most of the spot detection then decode approaches don't, at least the ones that are available like off the shelf in GitHub or published, don't include an explicit deconvolution step in the pre-processing. I do use one, but I actually find it adds a lot of issues, not help. And that's why I wanted to know in your case whether like either your pre-processing deconvolution or your subsequent kind of implicit deconvolution for separating the different parts of the code book, if I'm understanding correctly, the point of that second one, is actually helping you guys much. I just do I do a weak spot detection, a very noisy spot detection. I use a ransack filter to determine a subset of those spots, which actually are a good representation of the point spread function. And then I do Lucy deconvolution. And then that's my sort of pre-processing. And then I just do like standard difference of Gaussian detection after that. And I really have no idea how much value my deconvolution is adding. I definitely get more dense spot detection in areas where there's high expression of a specific gene. And that's my intention, is to try to like take these areas where I have tons of overlapping point spread functions and get the right or closer to the right number of spots that are actually there. But Richardson-Lucy, in my experience, is a really noisy, problematic algorithm. And so I'm just wondering, like in your experience, is it the order or is it the deconvolutions that you're doing that are making improvements?
I believe it's the order, but I could be wrong. And I mean, as you say, it is difficult to evaluate. We have a shared data set that is a synthetic data set that we would be super happy to share if you want to sort of compare your approach to what we have. And I guess it could also depend on what kind of microscopy approach you use.
No, I have maybe another question. So I always a bit like, so I mean, we also do a lot of this, right? And like one, or like maybe the main readout for us whether we do a good job or not is looking at this false discovery rate of these barcodes. But I always a little bit confused about this, because actually when I ask people how these barcodes are added, I get sort of not so like, I don't know. I mean, like, does that play a big role, or can one do this in a more or less clever way? And what do you think about this whole approach? Do you have anything?
Yeah, so either you just add every possible barcode that you could have. I mean, when you select the barcodes for what you're targeting, you should, of course, spread them out as much as possible to have them have as little overlap as possible. But then when you run your decoding, you could run it against the library containing every possible barcode. But and that would give you, a way of knowing how much is noise. But you could also alternatively kind of force, if you only include the barcodes that you know should be there, you can force all your signals to be similar to something expected, and in that sense sort of compensate for small variations. But then of course you don't have a way of knowing where to threshold for what is good quality and poor quality spots. And I guess you could do both. first do a with a complete code book, including every possible code, and then rerun it afterwards with only the codes that you should have, and then look at the difference in your quality metric between the two approaches.
But I guess common practice is to just add a few false positive barcodes, or not?
I don't know.
Ah, okay. So I thought that, okay.
I think we have gone for all.
Okay, interesting. Okay,
because
that's
the most common false barcode is the what we would call the Unicode, where you only have, you know, AAAA or something like that, because that's usually some auto-fluorescent artifact.
Interesting. Okay, then maybe you should look into this. Thank you. Okay, and Mark, you have a question?
Sure. Can you hear me?
Yes.
Okay. I was kind of interested in the last point because it looks like you were trying to infer some of the information we've learned from this labeling from like H&E, and so you're starting to think about your clinical pathology lab context. And I'm wondering if you thought about other ways to present it to that audience as well. I've seen approaches where you take fluorescence and make it more like the traditional IHC staining, which is usually kind of brown on top of HE or something like this. And if you thought about how do you present this data to a pathologist?
You're talking about in situ sequencing data in general?
Yes.
They seem to be quite happy seeing it as these little markers as we present them in our tool. I think that it hasn't really been a problem in especially, you know, in the if I show this again, you know, just the to be able to, I mean, here we can like quickly browse through and the different ways of seeing one thing at a time, and sort of having access to the morphology at the same time. They kind of like it. I don't know if that's answering your question.
Kind of. I'm just trying to think about how to kind of incorporate this into a clinical pathologist's kind of pattern recognition, because they...
Yeah. I mean, of course, I mean, you could present, I mean, dab staining, you could like simulate some kind of dab staining.
Right.
That would be quite possible, you know, just representing every dot as some kind of brown Gaussian. But I don't really know what the gain would be.
It's some consequence of the last thing you presented of mapping the you found similar looking clusters spatially in the transcriptomics data as you did in the H&E stain data. You train a network to predict the appearance of one image modality from the other. There are lots of examples of sort of multimodality synthesis networks in radiology. Collect a T1-weighted MRI, predict a T2-weighted MRI.
Right,
and it's fully supervised. As long as you have these parallel sections, you could train a network to do it.
Yeah.
I agree. I don't know what the application is really, but given what you've shown here, I think you could definitely train a network to do that successfully.
Yeah, no, definitely. I think what you need to be really careful with is that both the spatial omics data is very high dimensional, and you know, whatever approach you use for your learning is also extremely high dimensional. And then searching for similarities in two very high dimensional spaces with a limited set of data, you're really on thin ice. So and that's why we you know, in this approach here, we did the two, we did the clustering of feature space completely independent of the gene expression. And then once we had these clusters, that's when we search to see, are there any genes that are more highly expressed inside a pattern as compared to outside. So we really wanted to keep the two data, as I said, separately, because otherwise, I mean, you would very easily find correlations just because of the high dimensions. But in this case, this was a way for us to confirm that the morphological differences between the upper and lower half of this part of the tissue were sort of biologically relevant, since they also have differences in gene expression.
When you design your padlock probes, can you cover splice variants?
Yes, you can. Because, I mean, either you could, it depends on, I mean, you can either have that splice variant in the gap, but the gap can't be very long. But splice variants could also be found then by the first and the, I mean, the
Right.
beginning and
So
the
how
end
many-
of the probe.
How many
fitting
probes how many probes do you use per gene?
That depends on how long the gene is.
Okay. And then secondly, when you're doing a study of like 10 tissue samples or 8 tissue samples, are you using the exact same probe set? And
Yes.
are you fine? So those are all common to the single cell data from all of them.
Well, you usually, I mean, we have been in the most studies that we are involved in, we target, you know, a few hundred, 100 or 200 genes at a time. And because the more sequences you, or the more letters you decode, the more different things you can look for at the same time. But we usually recommend to first do a single cell sequencing to figure out what are the most relevant genes for differentiating between the cell types.
So you may be skipping outliers from some of those samples. If they're not expressed in every sample.
That's true. I mean, if we do the single cell sequencing on an independent sample, we might miss cell types that are unique for a given. That's very true. That's a limitation.
And you're attributing most of the differences in RNA levels to transcription because you're assuming that RNA decay is uniform. Do you think that's a safe assumption?
No, not 100% safe. But I think it's more safe than when looking at proteins, because I've been looking at quite a lot of tissue microarrays that have been stained with these, you know, this Polaris data and so on, where you have multiple fluorescent markers for different proteins. And the heterogeneity that I see there is enormous as compared to the heterogeneity that I see when I look at in situ sequencing data. And, you know, talking to pathologists and so on, my interpretation is that a lot of different things can happen from the time that you take out the tumor to when it's fully fixated. And the variations in that cause variations in protein detection at the later point. And it's something that is very, very difficult to correct for. And in there, I think that RNA is more uniformly degraded as compared to proteins.
I think that's the selling point to the pathologists, that RNA is a better marker than antibodies.
Well, my experience from working with antibody-based data is that it's very, very difficult to find a threshold for defining which cells are positive, which are negative. Yeah, I mean, we have been struggling a lot with that type of data. I think I even have a sample here. Let me see. that sort of illustrates the problem. So we also visualize tissue microdata with this tool. And I mean, you have beautiful stained material like this. And then if you point to, you know, like this cell, for example, is quite obvious that it's to the right here, you have the different stains in the different channels. But then for other cells, it's really difficult to tell if it's a positive or negative staining. I mean, just imagine thresholding all of these and saying that where do I have a positive signal and where do I have a negative one? It's really difficult. And then you have huge variations from core to core. Yeah, sort of banging my head in that world for a long time. I kind of prefer the RNA because I have the signals, both the shape and the intensity to sort of base my decision on if it's something that is true or just background.
So you guys were talking about validating and this false detection rate as a way of trying to validate. How many things did you find that would have been impossible to find based on the particular barcodes that were used? I don't know if this is possible from the sample preparation standpoint, or maybe it's already been done in like the methods papers, but you don't do it like per sample afterwards. But does it make sense to include a channel where there's no decoding? It's just that single channel indicates completely the presence of a gene or not. So more just the classic in situ hybridization.
Yeah.
And
So,
also barcode that gene, and that way for, you know, that takes up a few of your channels, but when you have the opportunity to stain for 1,000 genes, losing a dozen or so to a validation might be a worthy trade-off.
yeah.
That way you could say, all right, we barcoding found 100 copies of the gene. The more direct approach found 90 copies of the gene. Now we have an idea of what our error rate is.
No, that's a clever way of doing it. And I mean, what we also do is to have multiple probes for the same gene. And then you could see how often they overlap. One of the tricky things there is that the in situ sequencing is not 100% efficient. I mean, you have to have 2 successful hybridizations and a successful ligation of the 2 ends to have the rolling circle amplification. So there are many things. There could be many reasons to have a non-existent signal. Well, whenever you have a signal, you can be quite confident that you found the right thing. So that kind of complicates things a little bit also.
Yeah, I hadn't thought of that, but yeah, just barcoding the same target with 2 totally different barcodes is probably even better than just directly labeling something with a single, like a binary barcode.
I mean, what we have done in the this liver project is to go in and do fish or immunohistochemical stains for those findings that we claim are, you know, clinically relevant, just to not claim something we can't prove in that way as well.
That makes sense too. Yeah, because this is so high throughput and exploratory. I was very also curious about the section where you were saying you find so many things with these very multiplexed approaches, and you don't know which ones are actually relevant to the pathology. I'm imaging far fewer genes than you guys are, like the dozens of genes, but we still have that problem. And we're not looking for what's relevant to pathology. We're looking for what's relevant to calcium imaging, like what explains the neural activity that we looked in the sample before we sacrificed it. But we find that issue as well, where it's like really, really hard to, with so much data, to interpret like what's relevant. And so that makes sense. Big exploratory experiment, some clinical findings confirm with a more targeted experiment. We're doing something similar.
Cool.
Okay, so I would say let's wrap it up maybe. Thank you, Carolina.
Well,
very
thank you
much.
for inviting me and thanks for the question.